Let's catch up with this Glowworm Roadshow. Professor Kurt Krause from University of Otago joins me now. Hi there, Kurt.
Kia ora, Jesse. Now, everybody loves the glowworm, right? If you're from New Zealand, you've got to love the glowworm. Yeah. But it's actually really quite a unique insect. It has a number of secrets. Its glowing chemistry is unique compared to other glowing insects. There's so many things that we don't know about the glowworm, so we all get together in order to share information.
What's the point of the trip?
Well, it's about 11 New Zealand and Australian scientists and science communicators that are working their way up New Zealand, and we're sharing, exchanging information, advancing research, culminating in a hui at Waitomo Glowworm Caves, where we've sort of put all the knowledge together. I'm a biochemist, and what I'm interested in is the biochemistry of what makes the glowworm glow. But in order to really do what we need to do, we have to get together with entomologists who study the biology of the glowworm, geneticists, analytical chemists. We've got some science communicators with us, and it kind of takes a village in order to really begin to understand what to do.
I'm Kurt Krause. I'm a professor of biochemistry at the University of Otago.
I'm David Merritt. I'm an associate professor at the University of Queensland.
Hi, my name is Julia Casper. I work at Te Papa in Wellington, and I'm the curator for entomology there.
We kicked off our road trip in central Wellington, capital of Aotearoa, New Zealand, and home to some of the country's most urban glowworms. We trekked uphill in the dusk to the Botanic Garden for our first glowworm viewing fix.
But there's a bunch here, really. Yeah. Maybe 50.
So we're having just a good look at them.
Yeah.
Big thing about glowworms is they're not worms, right? They're actually flies. But the part that we see that we call a worm is really a larvae or sort of a beginning stage of development for the insect. And if you let the larvae grow long enough, and you have to feed it and house it and keep it comfortable, then it pupates and sort of forms like a cocoon-like structure. And then it emerges from this cocoon as an adult fly. And then the female fly lays eggs, and then the next generation of glowworms continues.
I've been in a cave and seen a glowworm flying and glowing at the same time. It was amazing.
Suspicious.
I didn't believe it. Just none of the Australian species do that.
Yeah.
Still in Wellington, the next day Julia gave us a tour of the invertebrate displays at Te Papa Tongarewa Museum of New Zealand, some of which were curated by Julia and her team.
So was that done with just macro lenses or something?
Yeah. Under the microscope, yeah.
Yeah, it's good. Was it dead or alive?
Dead.
Yeah.
How's it preserved?
In alcohol. It was actually quite old specimen.
Drosophila flicked in there. Now it'll come
But
around.
the snares were really done in the last second. Because,
Right.
And I have never really seen them until they were here.
Yeah.
So it took so long to get the worm right.
Yep. I love the way they turn around in the tube. Oh, yeah.
So they suck the blood out?
Yeah. They chew and swallow, so they're not sucking. Because they've got mandibles, so when you look at their gut contents, if you've ever dissected one, you can see all these little pieces of cuticle from the animal that they're feeding on, you know what I mean? So they're chewing up bits and eating them and swallowing them.
We walked along the road from the museum to visit Julia's office.
Thank you.
No worries. Wow,
These are some,
big female there.
but isn't it amazing?
So you reared these out?
Yeah.
I think ours is bigger.
What?
I'm serious. South Island one is bigger than that.
The Piri one?
No,
from the,
Nichols Creek.
But that was amazing because so it pupated, because Heine said when they are disturbed, right? They fall into, So they pupated the next day, and then a few days later, they emerged. It was just
Cool.
rapid.
That's the difference between New Zealand and all Australian species. New Zealand, the pupa hangs from one end. In Australia, they hang from two ends in a hammock.
She also gave us a special peek at the Hudson collection stored behind the scenes. The great George Hudson was one of New Zealand's pioneer naturalists. He showed the world that the New Zealand glowworm is a fly, not a beetle like the Northern Hemisphere fireflies. Throughout his life, he gathered the largest personal collection of New Zealand insects, now cared for by Te Papa.
Shelf, put up the straps, so now it's kind of earthquake safe, and we can access it. And since then, we have worked with that collection so much. So I can say I'm becoming a nerd now, because he's my hero. I mean, look at this. So Hudson was a troubled child in England, was bullied for his passion for nature, and he was quite sick often, and couldn't really cope with the school system, with the system in general, and was homeschooled by his dad quite regularly. His dad was an artist, like a glass artist, and they came to New Zealand. So his dad and himself and his brothers were already here, and he just thrived in New Zealand. He could follow his passion to collect bugs. So instead of putting the information, the metadata of the specimen on a little label that is pinned together with a specimen, he put all the information in those books. So he was absolutely an amateur, although I think he wasn't. He knew more than everyone else. All his books, everything he did was for the public. And that's why I wanted to have that collection accessible and want to involve the public.
So he's called...
No, see, he always put his Arachnocampa luminosus. He put the genus on top and the species at the bottom.
So he's calling them Arachnocampa. So these must be after the Scoozer's...
Or he revised his collection all the time.
So he would relabel.
Yeah.
Later that evening, we met up again at Central Park.
Wine and cheese.
And then stumble back through the glowworms.
Yeah.
Yeah, I think the difference to the botanical garden is if you stand here a while and get adapted to the absence of light, they are just around if you go deep in the bush.
They are amazing, aren't they? So close to the centre of the city.
Yeah, it's just incredible.
And you look up and you can see the stars.
Yeah.
Oh, that's pretty. Can you see that?
Yeah.
The glowworm's moving away.
No.
Yeah. Not a whole night.
Oh yeah?
2 or 3 hours. I mean, it's...
Don't you have to spend the whole night because you find out what they do at 4 a.m.?
Oh yeah, I'm sure it's very different to what they do with that.
Absolutely, it's those secret glowworms.
Yeah, their meetings and their little collectivization and infiltrate their ranks. It's day 7 and they suspect nothing. Sellotape's an LED to my nose.
So we were looking at fungus.
We saw one of these in the museum today. Hanging fly, it's called. It's living.
Like a
Oh yeah, I know what you mean.
like a charging capacity to it, where it takes on enough ultraviolet radiation after a time.
Could be the UV coming back to you, but I know what you mean. You know, it does look. It really does come up, doesn't it? So it's worth.
I'm not sure if it's just our eyes adapting.
Yeah.
The micro details of whatever your sensor seems to think is a priority. You can get like a nice little up close of their innards sometimes, and with the black light you can start getting ones. So that's the internal structure of its digestive tract and glistening.
They seem to all be glowing as well.
Yeah.
You know, so in the caves when things aren't going so well, they're not all glowing all the time. But these guys in the bush, they wait till the light turns off and that's when they glow their brightest, and then they just dim through the night. Michelle's setting up for one more photo.
And then we approached little cave, like more crevices caves. So we had to lie down on our board, and then like the ceiling was here, and then you're floating along the glowworm. Yeah,
That sounds nice. Yeah.
that was
You've got a nice story. Oh, gee, Julia.
Sorry. Okay, I see you all in my Tomo.
Count all 11.
The next day, we moved our base to Palmerston North. That evening's glowworm viewing was a little harder to get to.
How sure are you that there is only one species of glowworm in New Zealand?
Oh, that's actually a really great point, because
Hi.
traveling with me is Dave Merritt, who's probably internationally the most famous glowworm researcher in the world. And he played an important role in determining that Australia, they have 8 species. And so Dave, along with some scientists from New Zealand, Julia Casper, for example, from Te Papa, and Peter Dearden, and a number of folks, are looking at the genetics of the glowworm from New Zealand to see, could the New Zealand glowworm be more than one species. Right now, as you mentioned, it's thought to be one, called Arachnocantha luminosa, sort of the official name. But we're getting more and more evidence that actually the glowworms at Otago may be different from the glowworms on the North Island.
Is it a spider?
No, a glowworm.
Really?
Yeah.
Yeah. If you follow this like route down, you'll see the blue dot. Oh yeah, I did.
Way. How was that, Kurt?
That's challenging.
Wobbly afterwards, but
There's some logs on the way.
Oh,
We are going that way.
okay. I did a bit of rock climbing as well, but nothing bad.
I think when we were here before, we went through there. Yeah. And then it opens up past that area.
Yeah, so everyone turns their torches off. And you basically just got to stand in the water, staring into the chasm with your torch off and find them. There'll be quite a few hidden away in the crevices.
Okay.
how is it?
I assume you do the same as what we did and take them all back live and put them in a tray of water?
We did try that once or twice, but we found it was just easier to, I guess, not gather the muck up at the same time and just put them into tubes clean. And then we didn't have the water at the end. But I don't think it really mattered in terms of the specimens that we had right at the end when we were out and freezing them.
Did you freeze them in the field or wait till you got back to Dunedin?
I waited till we got back to Dunedin. Often I'd collect and then just pop them in the fridge until the next morning.
Okay.
And
Didn't eat each other?
you'd lose maybe one. They do like to eat each other. I think they're just practical.
Yeah, you just gotta eat when you can, I
They're
suppose.
pragmatic.
I was going to try and get you guys coming out. Alright.
I'd normally wear gumboots, but even they would have got filled with water and that.
So, yeah, there used to be a lot more.
Maybe they've had a big flood through here or something. There's a lot of debris perched up really high as if it's just funneled down,
So guys, there's another little glowworm spot. How far away from here, you'd say? It's just on the way back, and it's a 5-minute walk up a giant cliff and down a... No, it's...
Mountaineering equipment. I have started working on microbiomes of dung beetles.
Oh, interesting.
And she's been doing the dissections for me to get the DNA. And she's saving all the guts.
Yeah. See what they're eating?
And we can maybe get them... it'll come down. We can get them sequenced and figure out if we can use that as a way of telling where they're from, what they've been eating, all that sort of stuff,
What makes them glow, Kurt?
It's an enzyme reaction, Jesse. It's always a reaction where an enzyme oxidizes a fuel molecule, a substrate molecule, and it's usually a reaction of oxygen with this fuel results in this glow. So it's enzymes and fuel. So in my lab, we purified that enzyme and characterized that enzyme, actually traveling with some students who are purifying and characterizing it now. And we're trying to do a final identification of the fuel. So in biotechnology, you can use a luciferase as a method of identifying how how much of a gene is being produced, or even identifying, say, where a bacteria is. So we're hopeful that because the chemistry of the glowworm is unique, it might actually end up being behind a big scientific discovery down the road if we describe the chemistry here.
So look, what's that Mexican drink where you have a worm in the bottom of your alcohol? What's the word?
Tequila?
Tequila.
The road trip continued onward to Taumaranui. Kurt was interviewed by Jesse on Radio New Zealand National before we headed out to Kakahi for the evening.
How's the road trip going?
Oh, it's been great. Actually, it's been fantastic. We were in Palmerston North yesterday, and now we're in Taumaranui. It's been great.
Oh, look how much he's moving. Wow.
Can you hold him up?
I think he's a bit small.
Wow. It's a slug, yeah. Because it's got antennae.
Let's have a look at the slug.
I'm not a very good biologist, but I know that.
Hello, slug.
This time there were lots of other creatures to see as well, including slugs, spiders, a very well hidden paradise shelduck, and a lot of glowworm food.
Yeah, I can see them.
Day 5, we reached our ultimate destination, Waitomo.
And Waitomo is probably, I guess, the spiritual home of the glowworm in New Zealand, right? And so we wanted to get together with people from the Ruapuha Uokawa Hapu Trust and with the Kaitiaki at Waitomo, who's Shannon Corkill, and share and partner together in order to make sure that we're doing things right in terms of our glowworm research.
We were given a special tour of the Ruakuri cave and its glowworms.
And this is disguising an entrance that we built, but to connect to a cave passage which is on a level a bit lower. Can be a bit intimidating coming into this view. Normally the lights are off. So I guess it helps you understand how important this place is to more than just visitors, more than just people that are looking after the conservation side of things, but also from a spiritual, cultural point of view, this cave is quite significant. And so it's a privilege to be here as well.
So up on the left, on the muddy bank there, we've got a whole heap of monitoring equipment. The major concern here is CO2. Quite a conservative threshold has been set at 2,400 parts per million, so we don't want to exceed that threshold. At extended periods of time, it can lead to condensation corrosion.
There we go. There they are.
The snares are showing up nicely.
Yeah, they are. Yeah. I'm just being a bit protective of the wall behind me here, so the glowworms, these glowworms will be doing so well that they actually hang their fishing lines over the walkway. Just remember not to move too quickly, don't create a breeze, don't breathe on them if you can help it, hold your breath. That was one good thing about COVID, people wearing masks, it stopped too much breathing going on.
There's a pubis there hanging vertically, a lumpy one.
It's there, it's an empty one. It's there, yeah.
They seem to do well in environments that aren't really, really wet. But I'm fascinated by why it's taken that many years for these colonies to get where they are. Maybe it's just trial and error if some stray female happens to lay some eggs.
That's what I see, yeah. And this time of year when you see the little hatchings, oh, right, there's glowworms over there. I wonder what will happen with that.
You notice how the bottom droplet is always heavier than the others. It sort of keeps the lines from
like a weight
moving around.
Yeah, a sinker. And then sometimes it's a even bigger and darker coloured droplet.
Right.
Which I believe can be waste products.
Well, they poop out. They stick their bum out of the horizontal line, and then just on video that they just defecate and it drops. So
Just drops straight
they
out.
don't hang them on necessarily hang them on the lines.
Another one further up to the right. Yeah, just up here. So I'm going to use my shadow to show you. So I've circled a little empty case there and the little eggs just beside it. So it's a small cluster right at the tip of my shadow there. And then hanging just in front of it is...
God, I would never have thought that's an egg cluster.
Yeah, they blend in quite well.
Day 6, the glowworm hui at Waitomo.
One of the best ways we feel maybe to protect the glowworm is to learn as much as we possibly can about the glowworm, right? I actually have a sort of a lifelong interest in bioluminescence, and when I was in graduate school years ago, I got introduced to working with glowing bacteria and ended up working on glowing bacteria and then switching over to working on glowing dinoflagellates that grow in the ocean. When I shifted to New Zealand about 16 years ago, we learned that we had glowworms in a grotto about 5 minutes away from where I lived, where the chemistry of how they glow, the nature of the enzyme that makes the light and the nature of the fuel that makes them glow hadn't been worked out yet.
At Waitomo, we operate under 3 core values. Manaakitanga is to provide great hospitality to our customers. Kaitiakitanga is, in essence, my role in what we're sort of all trying to achieve by paying an admission ticket to go into the caves, which is to protect and preserve those beautiful spaces. And Whanaungatanga is all about forming relationships and looking after our crew.
So this tracheal layer is really closely associated with the Malpighian tubules, which really associates that access to oxygen as a critical factor in glowworm bioluminescence. And this is a micro CT scan of the light organ. So you can see these 4 finger-like structures. These are the modified Malpighian tubules. They're much more swollen than normal, and this is where light is produced.
And actually it was kind of surprising what we found out. Glowworm luciferase is the same shape, roughly the same shape as the firefly luciferase. But they use different luciferins. Like the little molecule is entirely different. From a molecular convergent evolution point of view, that's quite intriguing.
Figuring out the glowworm luciferase is essential to figuring out how it functions. This is easy enough in theory, just go and do the science things and figure out what it looks like. Easy peasy. We've encountered this one problem, and it's a problem called protein insolubility.
And here, the evening increased, just dimming down, and then some rain started, and bang, they just shot up their brightness. And that's 4 different occasions there to show that. How? I think is they detect the vibration of rain droplets on the substrate, on the forest floor. They're very sensitive to vibration, as we saw today in the glowworm cave. Loud noises, bang, they brighten up like crazy. Why? I think it's possibly because flying insects that they feed on, they may be activated by rainfall as well. This is a time-lapse shot out of Waitomo glowworm cave. It's captured at 1 frame every 30 minutes. It's played back at 25 frames per second, so each day is occupying about 2 seconds in this video. You can see a cycle there, they're brightening and dimming every day. And so in a cave, there's no other clues as to what time of day it is, but they maintain a cycle. They stay synchronized, and it's a precise 24-hour cycle, so they're registering off each other.
The enzyme is made out of a long string of amino acids, and one end is called the C-terminal and one end is called the N-terminal. So this is the C-terminal end, and it is aligned to the firefly luciferase.
Again, thanks to the pandemic, Dave couldn't come here and ask me, hey Julia, can you collect glowworms for me from across the country? So it's me, and you can see the camera is quite blurry, and the lens, because it's super wet and muddy in there, and yeah, you have to squeeze through some bottlenecks and wade through water to here, and yeah, it's quite nice. So, and I do rear the glowworms, or try to get them to adulthood from all the places, because we want the voucher specimens in the museum, because there are barely any it's really hard to get the adults. So apart from that genetic and taxonomy project, it's really good to have the voucher specimens from the different places. So and that is one of mine that emerged.
It'd be good if you have questions about what you heard today, or also if you have comments or recommendations about how to keep this going.
If you look at the catchment, say upstream from the Glowworm Cave, how much of that area is likely to be forested?
John may be able to answer that better than I can, but certainly on our farm I know that all of it was felled for the Whitesmore catchment. I've possumed all the way down through the Whitesmore catchment, and I would say might be lucky to have 20%.
A while back, it was not a very good picture, but it actually shows you the different colour. The pepper glowing is actually quite a different colour.
Yes, wow.
So it's a bit hard to. It's kind of more of a yellowy-green colour.
Yeah. It's really good to see all the scientific development and investigation done. It's great to see some of the dots connected.
We've had phenomenal participation from everyone, so it was a really awesome opportunity to all come together and talk about our titiwai and our caves, our taonga, and more specifically, and how we can care for them. Yeah, thank you all, and hopefully we can do it again sometime. But that was all for me.
Thanks for sharing your passion with us today.
Thanks so much for asking us.
Yeah, you'll always be welcome. Kurt Krause, Professor at University of Otago, talking glowworms.